Designing Materials by Exploiting Water Interactions is my title today. And I would really, and I'm pretty sure that it is self-evident for this audience that the biological systems, they always exist in aqueous environment. So that means that these water interactions are really at heart of the biomaterials engineering. If we consider all the plant cell wall functions, they are actually based on water interactions. If we consider the main components, cellulose and hemicelluloses especially, they are the most hygroscopic and hydrophilic components you can actually imagine. And if you think about the applications, water is very often used as a detrimental feature. You try to get rid of it once producing paper, and you lose very much of its performance when it's in contact with water, even with moisture. But on the other hand, these water interactions, they truly define the behavior and the mechanisms. So when you reassemble these lignocellulosic building blocks, they always have the inherent response on moisture and water. And that is actually very inspirational. You could actually, and could you actually draw inspiration from this moisture and water responsiveness for applications? And I think that hygroscopicity, it brings a lot of performance. It brings anti-fouling features. Water transport functions can actually be utilized, for example, in membranes, which I'm going to show you today, and also similar fashion, the anti-fouling features. And I think that one of these very intriguing parts, again, self-evident, they are biocompatible, but also how could you actually embed living materials with lignocellulosic or nanocellulosic systems and enhance the viability and improve the cell performance. I think that these type of examples I'm going to show you today, they really kind of show the importance of water in materials engineering. And I would like to remind you on the research themes we are dealing within the FinnCERES program. And we are divided into fundamentals, where actually the water interactions and advanced characterization, which is part half of my talk today, that how we are actually looking at the water interactions, and especially the theme called clean air and water, that is very much dedicated to translating these water interactions to something useful. And there we actually do this efficient capturing of valuables, rare elements and pollutants, and these water purification systems, and also the monitoring of water systems, environmental monitoring. It's very good idea to have them in a very in that type of stage that they are very hygroscopic. So grand targets in FinnCERES is really to understand the interactions and also the architectures that enable us to produce, manufacture, construct selective and sensitive capturing of components. And first, I would like to go through the very systematic approach, very fundamental approach, how we have been investigated the water sensitivity of various nanocellulosic materials. And we have been doing that via interfacial investigations. We have been monitoring the water vapor uptake of different types of cellulosic materials, and these water interactions, they are actually very seminal topic among the biomaterials research. And in literature, and they have been studied extensively using bulk methods like dynamic vapor sorption, and also there's quite much of the literature on specific models which you can fit into water and vapor sorption. And when it comes to nanocellulosic building blocks, both fibrils and crystals, the moisture sensitivity is actually one feature which differentiates those nanomaterials from many other nanomaterials. So once you can actually tune the local geometries at nanoscale, which by moisture by water, where you could actually estimate or foresee applications in sensors, diagnostics, membranes, and biocatalytic matrices. And we have been working on this surface-sensitive approach to explicitly reveal the interaction of nanocellulose, of nanocellulose with water vapor. And the next slide takes a little bit of time to appear because it contains so heavy information. And it also dates back some years already before FinnCERES times, when we actually started to introduce this surface-sensitive approach, combining acoustic method, QCMD, quartz crystal micropulse, with optical method, spectroscopic ellipsometer, to really look at the vapor uptake behavior of these cellulosic systems. And the most simple system were the ultra-thin films of cellulose nanocrystals. And we were, in the work of Elin Inivara, we were able to evidence that these 3 molecular layers of water envelope each CNC in the network. And this means that there is this 1 nanometer layer thickness of water on CNC layer. And I think that kind of gives us impression that what is the resolution and preciseness of this combination of surface-sensitive methods and ultra-thin films. And going towards a little bit more, I would say, more complex system, we looked at the vapor sorption of cellulose nanofibrils, and again, combining these 2 optical and mass detection methods, we were actually able to gain precise insight on the changes in physical attributes. And we were actually able to fit models, these simple models, Langmuir model in dry conditions, in semi-dry conditions, the Florey-Huggins model, and in very high relative humid condition, the clustering model on how the cellulose nanofibril network is reacting on increasing relative humid conditions. This approach enables us for further evaluation of sorption kinetics, and also we are on the way to really detect the diffusion and permeability coefficients of nanocellulosic materials. And putting that into an application, these coefficients and this understanding is highly interesting and important when we are talking about, for example, packaging barriers or membranes and air purification filters, for example. And this is now the latest advancement, what we have been doing within the FinnCERES program together with Eero, and also we shared a postdoc, Chu Chung Meng, who actually introduced the hemicelluloses in this system, which has so far been purely cellulose-based system. And in this approach, we were able to actually, with this thin film approach, we are actually able to increase amount of xylane on top of the cellulose materials, and then again monitor the moisture uptake of these systems, again by QCMD and SPR. And with this ultra-thin film approach, it was actually noticed that there are this type of minimum value for water uptake, which is not a result as expected. You would probably expect that by increasing the ratio of amorphous hemicellulose, you actually increase the water uptake of the material. But there is this type of minimum value detected. And also this very same trend was also noticed with fibers, with bulk method, looking at the dynamic vapor sorption. And this is very often claimed that when you are dealing with ultra-thin films and surface-sensitive methods, does it have anything to do with realistic conditions, with the realism? And this is a perfect example that actually this ratio of hemicellulose with crystalline nanocellulose shows that we again follow the similar type of trend of vapor and water uptake. So there is a kind of the reason that the ratio of amorphous and crystalline material in cell wall composition actually seems to define the moisture uptake behavior. And very wild, not proven, kind of tentative conclusion is that can this be actually linked to plants' natural moisture management, for example, in wet and dry conditions. And this is the place where we actually intend to connect with plant physiologists and biologists to better understand. And we have started the discussions already. Paul Dupré is here today also showing the role of water there. And also Anna Kärkönen from Luke, we are now kind of broadening this, that can we actually better explain what is really the role of hemicelluloses here in the moisture management of the plants. So, as a conclusion so far is that we can probe these water interactions with nanosensitivity. On top of that, we can model the amount of and location of water as well. That part we are doing in collaboration with Jukka Ketoja and Antti Paajanen in the frame of FinnCERES. So we combined also the molecular simulations, and what you should kind of notice here that actually the length scale for the models and the model systems are today on the similar level, which should help us. And we actually have quite ambitious research questions that can we actually use the water molecules to probe exact size and shape of the elementary microfibrils, and can we reveal the role of hemicelluloses, for example, in fiber swelling, because these questions are both of those questions are under debate at the moment. It is not clear. And can we actually use these water molecules, because we can go so high preciseness with molecular simulations, that can we give some kind of contribution also on this discussion. So my second phase of my presentation is that how can we actually turn these fundamental discoveries and how can they be applied in really in the application related work. So I showed you that we do have these very interesting findings on pronounced water uptake ability of nanocellulose, moisture responsive structures, and also we do have very many strategies to enhance wet strength, because when applying the water interactions in materials engineering, it doesn't mean that we completely forget that it's also detrimental, that you need to improve, for example, wet strength in many applications. And here is the list on different type of application areas, and today I will share some results related to water purification, capturing of microplastics and anti-fouling membrane systems, and also the how we can prepare or manufacture the bio-based sensor elements using the high hygroscopicity, and also very short example on this carbon dioxide conversion for chemicals production. So first, bio-inspired materials. How could we capitalize the water interactions? And the first example I do have here is related to anti-fouling ultrafiltration membranes. And just a quick reminder, membranes are selective semi-permeable barriers and used in industrial processes for water purification, for example. And what comes to nanocellulose, cellulose nanofibrils, very many of its characteristics are useful in membrane science: large surface area, high hygroscopicity, nanoporosity, and also the film forming tendency. Putting these together, are we able to prepare nano-enhanced membrane materials? And this is kind of the latest example where we have actually been textured the commercial polyether sulfone membranes by using the combination of two nanofibrillar grades. the idea there is that can we actually tune the porosity and strength of the membrane surface coating material by selecting this type of combo of cellulose nanofibrils. And in this blueprint, we have actually been doing this at VTT Sutkola in pilot scale, showing this commercial membrane coating, having the wet strength and porosity tailored, improve the interfacial tailoring by adhesion by tailoring, by anchoring the nanocellulose combination using the cationic PEI polyelectrolyte, and improving the wet strength by acid-induced interfibrillar interactions. So this is, I would say, really like doing science in pilot scale. And I mentioned that these nanocellulosic materials, they really provide this anti-fouling property because of the charge and also because of the high hygroscopicity. And this is an example where we have compared the commercial PES membrane and these tempo-oxidized nanofibrils and mechanically disintegrated nanofibrils, and how the adsorption of bovine serum albumin, very well-known model protein for these organic foulant studies. And how you interpret this figure here is that higher negative change in frequency, more BSA is adsorbing on the surface. And as you can see, this commercial PES membrane, it actually, the BSA is adsorbing on this commercial membrane very, very much. But if you coat the PES membrane using either or nanocellulose grades, there is no change in frequency, which means that there is no BSA adsorption taking place. So we can claim that this anti-fouling property is directly evidenced. We also did with this similar membrane materials the adhesion and biofilm formation of E. coli bacteria. And these semi-images quite straightforwardly indicate that on PES membrane, these bacteria is really kind of, the bacteria is really colonializing the surface. But this tempo CNF surface, this one here, you can actually see that there is no E. coli detected on the surface. Intriguing again is that why does it adsorb or colonialize CNC surface, and also the other finding was quite surprising that actually this bacteria can degrade the CNC surface. But definitely tempo CNF surface was intact by the E. coli, which is good antibacterial performance shown here. And filtration performance of these nanocellulose-coated membranes, we see the high rejection of this protein, which is a good thing. But if we compare the rejection versus permeance on commercial PES membrane, shown here, we can see that we lose when it comes to the permeability and flux due to the very tight nanostructure. And here I think that again, we are dealing with the materials, how the porosity is kind of formed, and how strongly actually these nanocellulose networks are binding the water, because we know that there is a ratio of bound and free water every time when we do have this type of nanocellulose or cellulose network or cell wall structure. So I think that the root reason to tackle or to better understand is that can we actually manipulate the balance here, and via manipulating the porosity, how large pores we actually need in order to have more free water, better permeance, because we actually do have already the really nice rejection levels, and we are dealing now with the membranes with the pore size of few nanometers. And if we would like to go to higher porosity levels. That's the kind of the feature we need to address, is the balance between these different fractions and physical state of water. I think that it is quite evident that why these nanocellulose layers are good in membrane application. High hygroscopicity, hydrophilicity is a good thing, but today we are addressing in FinnCERES program that how to improve really this membrane flux and permeability. And then I will have a little bit more concrete example that where can these membranes be actually used. And we have shown that by using the hygroscopicity and by using these water transport functions provided by porous nanocellulose networks, we can actually convey and capture colloidal particles inside the material. So the work aim has been to introduce this early stage capturing of nanoplastic particles, and also to provide tools for assessment, because they are lacking as well. Also provide tools for reduction and removal, and on top of that, we would really like to understand the role of surface interactions in the capturing process when we are dealing with this quite inert nanoplastic particles and nanocellulose networks. And this picture actually shows, from the quite recent review paper, what is the gap there in understanding this nanoplastic and microplastic management. Even advanced wastewater treatment plants, they cannot deal with the nanoparticles. So the sampling with this smallest, tiniest fraction is impossible. And the same applies with the methods to detect those, that they are not existing. And what we have done, we have introduced this cellulose network, nanocellulose network, look at the gel function, water transport performance, and can we actually capture nanoplastic particles. And with this microfluidic setup, where we use these fluorescently labeled particles, you can actually see that those fluidic channels were in the presence of nanocellulose. You can see that they are quite well trapped once the water is kind of conveying them inside the gel structure. So the particles are really accumulated inside the network. And we also wanted to look at the mechanism a little bit more precisely, how much there is role from surface interactions, because our main hypothesis was that it is really this water transport which plays the role. Again, we looked at the adsorption via QCMD. Again, higher negative change in frequency indicates more adsorption of the particles. And if you look at the tempo nanocellulose film, there is no change in frequency, so we can conclude that high hygroscopicity is actually assisting the capturing process. With native CNF and regenerated CNF, we can see also some adsorption of the particles, kind of further helping the entrapment of the nanoplastics. So our conclusion here is that amphiphilicity of these nanocellulose systems and cellulose systems may actually generate the favorable interactions. And just to conclude this part, that it is quite evident that this hygroscopic nature, nanoporous nature, large surface area are the features which are helping us to capture colloidal materials. And actually this finding was noticed by The Guardian as 1 out of 4 innovations, promising innovations on tackling the microplastic challenge. And then I would like to move forward how we can actually use this water, role of water when we are hybridizing the nanocellulosic materials with carbon for electrochemical detection. And just shortly, here we intend to prepare the perm-selective membranes for selective and sensitive monitoring. This is the work done together with Katri Konttori from our side, and then Jari Koskinen and Tomi Laurila from Aalto's side. So they are the electrochemical experts, and we provide the nanocellulose grades, and also the dispersing these with electrically conductive carbon materials. So perm selectivity and electrochemical features are those which are needed. And by introducing nanocellulose, we actually provide hygroscopicity and again the anti-fouling feature. And can we actually use this type of platform for biosensing and for environmental monitoring? And an example here is that where we have compared sulfated cellulose nanofibrils and sulfated cellulose nanocrystals, hybridized those with carbon nanotubes, which is not trivial at all if you think about how different those materials are. But actually nanocellulose is able to provide this type of dispersion ability also for the carbon nanotubes. And here the electrochemical activity has been followed by using the, I need to check a note, it has been used by an electrolyte, hexaamide ruthenium trichloride, which is very typical electrolyte to probe the electrochemical performance of the materials in cyclic voltammetry. And here you can see very much response when it comes to the fibrillar network compared to CNC network. And again, we are dealing with stronger swelling, which improves the response, higher charge density, and also both of these materials, they show excellent stability over wide potential range, which are all good news. And this work is ongoing in the frame of FinnCERES in the collaboration, and now we are going towards the environmental monitoring and water purification systems with this hybrid approach. And my last example, which I will only very shortly show you, is this nanocellulose-based cell factories, where we are actually keeping up the cell performance by embedding it to the biocompatible nanocellulose network with high hygroscopicity, again, high porosity, which is kind of enabling to sustain the life and the performance of living cells. And here the idea is that these cells are embedded in a cell factory system, and in this case we are talking about an immobilized cell factory system where the cells are actually doing something useful. In our case, they are producing chemicals using solar energy and atmospheric carbon dioxide. And by embedding these cells to the nanocellulose matrix, we can see that both the ethylene production is increased, so the performance of the cell has been increased, and also the stability and mechanical performance has increased, because the reference system based on pure alginate, which is very often used matrix for this type of immobilized systems, it degrades, especially when we are talking about these very challenging submerged production conditions. But I'm not going to talk about this any further, because we do have the real expert on solid-state cell factories today here as an invited guest, Dr. Sergei Kosorov, who will give a more detailed talk on the topic. And there is also a poster by Ville Rissanen and the algae, VTT algae group in collaboration with the Turku University. So please visit the poster in the poster session later today. So with these words, I would like to welcome you to the new era of bio-based materials. Thank you.